So this is our first year at this conference. So thanks for having us. So the SymBio, introduce it real quickly. Biology has evolved an amazing array of diversity, and I think what we're asking is maybe there's a few good ideas out there that evolution just hasn't gotten around. And actually, the products that the Discovery Lab was looking at, like in grapes and such, it's actually a pretty good segue into what we want to show you. And so anyway, Natalie and Maya and Leah had a really good idea that they call Christmas beans, and well, I'll have them tell you about it.
All right, Natalie, can you share our suite?
Yeah, I've got it. Okay. Hi, everyone. So we were in synthetic biology, and our project was all about creating transgenic soybeans that will produce pinene. Pinene is the compound that is found in pine trees that gives them their distinct Christmassy smell. So pinene is a type of compound called a terpene, and terpenes are these unsaturated hydrocarbons that are really known for their aromas and flavors. So you can see off to the right, like a ton of these examples. Some people asked us in the comments on our little 5-minute presentation. So pinene obviously is known for smelling like pine, and it's really used in the perfume industry. Also, it's used as a solvent, it's an insect repellent, and it can be a biofuel. And then there's also some medicinal uses that still need a lot more research, but there are some anti-cancer uses and some anti-inflammatory uses. So why are we making beans that produce pinene instead of just getting it from a tree, right? So the problem is mostly environmental. So pinene and other monoterpenes are extracted from plants. So to get pinene, you obviously have to cut down these trees, which contributes to deforestation, and we don't want to contribute to that. So it's a lot easier if you can just grow these kinds of compounds like pinene in a field that's already used for other crops. And soybeans have a lot of research on growing efficiency, basically. And obviously, it's a smaller environmental impact if you're using fields that are already in use and not cutting down forests. And additionally, other studies have used E. coli or yeast to synthesize terpenes artificially. And those kind of work, but we think we have something better by using the soybeans. So the terpenes are synthesized in the chloroplasts of plants. And obviously E. coli or yeast don't have chloroplasts. So that would be another step, another thing to put inside of the E. coli genome. So soybeans are a plant, so they already have this MEP pathway through which the terpenes are synthesized. So they have this molecule called GPP, which you can see here. And GPP is the precursor to the pinene synthase. So all we have to do is add in this one sequence of pinene synthase, and then our beans can start producing some pinene.
So how we're going to do this, or what we've done so far, is we've made this really, really big vector, and basically it's going to go into the DNA of the soybean plant, and then the plant will use that to create the pinene synthase protein, which it does not have originally. It'll send that protein to the chloroplast, and then the pinene synthase in the chloroplast will make us some Christmas juice. So the 15,000 base pairs, in terms of synthetic biology, like that's a beefy plasmid. It's really, really big. And when you get bigger vectors of plasmids, you're introducing a couple more risks. One of them being you have lots and lots of recognition sites for restriction enzymes, and they're a little bit less stable. So we've had to come up or use some pretty special techniques to get around that issue. So one of the techniques that we use to get around the issue of having so many recognition sites within a big plasmid is by using Golden Gate restriction enzymes. So if you're not already familiar with what a restriction enzyme is, it's basically an enzyme that attaches to a very specific sequence of DNA. And most of them cut right in the middle of the recognition site. And then they form sticky ends, so you can kind of glue different pieces in. Golden Gate restriction enzymes, though, are super special and exciting because the recognition site is not the same as the cut site. And what this does is once the new plasmid is created, the restriction enzyme recognition site's no longer in the plasmid, so it's irreversible. And then you also have custom overhangs. So you can do any 4 base pairs, and this is really cool because it allowed us to put in multiple genes at once into the same vector.
Yeah, and so here we have the steps on how we use Golden Gate. And so the first step was assembling our pinene synthase DNA, and it actually had to come in 2 parts because it was such a big region. And then the second part is really the key to synthetic biology, and that's adding organism-specific promoters and terminators. And so in this case, we use soybean promoters. But the beauty in this is that if you have that promoter, you can change what the coding region is. So we use pinene, but the other group used humulene, and you can really change it to be more complex biomolecules in the future, but we're just using this as a simplified model. And then the third step is adding this fluorescent tag. And this just kind of works as like an indicator that we have successful implementation of our gene into the soybean. And so we wanted to test for successful assembly, and we did this in 3 ways. The first being DNA sequencing, the second being step-specific antibiotic resistance, and the third being electrophoresis gel. And so on the right here, we have one of our plasmids. This is the second to last step. It has our promoter pinene chloramphenicol antibiotic resistance and our terminator. And we were able to use BSA1, a restriction enzyme, and predicted cut sites to determine if our assembly was successful. And so if we had an unsuccessful assembly, we'd maybe see too few or too many fragments on our gel or something like that. But here we see proper fragment size corresponding to the predicted BSA1 fragments. As for what's next, we currently have our plasmid without the fluorescent tag and with a different antibiotic resistance, and it's an E. coli. And so we need to add the fluorescent tag. And then after that, we want to put this plasmid into the soybeans. And we're doing that by kind of using a middleman, and we're able to insert this plasmid into an agrobacterium, which can insert our DNA into the soybeans. And then after that, we need to test for pinene synthesis. And so we'd have to extract the pinene using CO2 extraction, or, my goodness, distillation. And then after that, we can analyze how much pinene is in there using gas chromatography. So then in the future, we could use this method, as previously mentioned, for a variety of more complex or broader use biomolecules. Lastly, we'd like to thank our professor, Dr. De Decker, as well as Simon Kalmus and Michaela Vassity for their guidance and instruction in the lab. We absolutely could not have done this without them.
Stefan, would you like to ask a question?
Yeah, I have a quick one. So frankly, is this actually safe? Because pinene's incredibly flammable. So you're basically creating kind of, correct me if I'm wrong, more flammable soybeans.
So terpenes are really volatile. You're right. That's definitely what makes them smell pretty good. The answer is we don't think it would be more flammable. Green things don't have a tendency to burn. I guess maybe toasted up soybeans.
I'd also argue that it might not be in significant enough amounts for it to make it like extra flammable.
Colin?
Yeah, I was going to ask, going more along the lines of making it more flammable, I guess, is in bacteria, when they use the bacteria and yeast, when they use those to create proteins, oftentimes they have a promoter that they can increase the expression of the gene with by introducing a molecule. I was wondering if you guys were trying to introduce that in your model organism, or if you were just going to let the plant naturally make however much pinene it did make.
We have a method for this, but our lips are sealed.
Okay.
I think, yeah. Email Dr. Dedecker.
Yeah, sorry we can't get you a super clear answer on that. We believe our professor is actually trying to get a patent on the exact system in which we're doing that.
Yeah, so it's a secret for now.
But you are planning on doing that?
Maybe.
Okay.
Perhaps.
Good question. Would these soybeans be edible, or like would you not want to eat them? And would they taste kind of like pine needles? I'm sorry, that might be a stupid question, but
no, that's actually a really good question. With our system, they would be edible for a part of time, but we wouldn't be like selling the beans on the market. It would be just the final terpene product.
Okay, another thing, when it comes to like planting the beans, I'm sorry if these questions are like super off topic, but if you come to planting the beans, right, is it possible that they would then breed with other beans in however way plants do that, and then spread this genetically modified terpene producing bean to places? That's way further out than you guys are going, obviously, in this lab, but like there's a lot of questions when you start talking about changing.
I would assume that we'd be like really careful about the beans and only keep them like in a specific area. Like I'm pretty sure that's what's going on now with the CBD beans in the same like with the same lab, but I don't know, maybe there's a dystopian future where all beans taste terrible and piney.
I will say I think that's
I'll actually interject here real quick. So
Okay.
soybeans are actually pretty good about this because their pollen's quite heavy, and so you only need like a 5-foot barrier around soybeans before you made transgenic beans everywhere.
The beans are being socially distant.
That's fantastic.
Exactly.
Wow.
We have time for one last question, Louis.
Yeah, thank you. Super great presentation, guys. So I was, since the plasmid is so big and you're using like another medium to transform it into the soybean, are there then any issues associated with possible mishaps with the incorporation of the DNA into the soybean, like possible mutations, or how do you guarantee the efficacy that the transformation is going to work correctly?
So up until now, we've ensured that like our vectors are accurate through antibiotic resistance on plates, gel electrophoresis, and then also we did run a Sanger sequencing. When it's in the plant, it will have the fluorescent tag if it was successfully transferred into the soybean genome.
And along the way, we are using like stabilized special E. coli cells because I think not all strains of E. coli could actually take a plasmid this big. So we have special ones.
Gotcha. Thank you.
Thank you for your question.